All right, we are live on Facebook.
Okay, I'm gonna go on mute.
All right, good afternoon, everyone. Welcome to Beaty at Home today. It is August 12th. So our Beaty at Home today is about bryophytes, fungi, and lichen. So today's presenter is Dr. Karen Golinski. She is our Beaty Biodiversity Museum Collections Curator for the Herbarium for the bryophytes, fungi, and lichen. So if you've not been to the physical Beaty Biodiversity Museum, I can tell you a little bit about that. The museum and UBC, are located on the traditional, ancestral, and unceded territory of the Musqueam people. So we encourage you to learn more at musqueam.bc.ca. So this image you're seeing here is actually the Musqueam flag. It was raised at UBC in February 2019. And if you wanted to see a map, so here's the city of Vancouver. We're right up here. We have just a few weeks ago reopened to the public, so we look forward to seeing you socially distantly at the museum on site. So yeah, here's a shot of the museum. You can sort of see our blue whale skeleton there. That's the blue whale skeleton. Not a bryophyte, but one of our specimens at the museum. Yeah, so we're part of the Biodiversity Research Center. So this is attached to the museum. It includes the museum and the research center itself, where over 50 faculty and their graduate students and postdoctoral researchers from departments across UBC work together on all kinds of biodiversity, contemporary research, ecology, evolution, taxonomy, communities, conservation, climate change, mathematics, and genetics. So here are some of the collections. We have 6 collections within the museum. We have the tetrapod collection. There's a platypus skeleton there representing them. So these are 4-legged vertebrates. Also representing the animals, we have some of our marine invertebrates, the yellow shell there. If you look at the purple butterfly, that represents our entomology collection. So that's insects and other terrestrial arthropods like arachnids. We also have our fish collection, and of course our fossil collection joint with the Pacific Museum of the Earth. But up top right, the green one is our herbarium, which contains basically more diversity than all those other animal ones combined. We've got all kinds of algae, we've got land plants, including mosses, vascular plants, fungi, and of course today we're focusing on the bryophytes, fungi, and lichen. So I'm going to pass the time over to our special guest, Dr. Karen Golinski, who will tell us a little bit more about that collection in our museum, our, well, bryophytes, fungi, and lichen. So I'll pass it over to Dr. Golinski.
Thanks, Vincent. Hi, everybody. I'm really excited to be here and saying hi to everyone today. I just started the position of the Collections Curator of Bryophytes, Lichens, and Fungi in May. I came from Washington, D.C., where I've been living for the last 6 years or so, and I've been working on the bryophyte collection in the U.S. National Herbarium at the Smithsonian Museum of Natural History. So I was really excited to get back to BC. I'm from British Columbia and actually did my undergraduate at UBC in landscape architecture, and then went on to do my doctorate at the University of Victoria. So I am taking over from Olivia Lee, who's the longtime manager of the Bryophyte collection. I think I can share my screen and show you maybe a picture of Olivia and myself when we were on Haida Gwaii a couple of years ago. There we are at a place called Takakia Lake. Can you see it? So Olivia and I have been friends for quite a long time because I was coming over here while I was doing my doctorate to work on specimens in the herbarium. And in fact, we just got together yesterday out in the courtyard here at the Beaty to do some filming and kind of reminiscing and talking about changes over the last 10 years or so. So the bryophyte collection at UBC is very large. It's one of the largest in Canada and Western North America, and there are about 327,000 specimens in our collection. Those are dried collections that are made when people go out into the field, and they represent a lot of diversity from the province and actually all over the world because we have a very active exchange program. Also under my domain, I guess, are the lichens. And we have about, I think, 45,000 lichens in the collection. I'm not a lichenologist, but we do have collections curators, Trevor Goward and Curtis Bjork, who are co-curators of the lichen collection. And we have about 45,000 or about 25,000 specimens of fungi. Those are the mushrooms. And our curator of the fungi is Mary Burby. She's a professor here at the museum. So bryophytes are everywhere. I want to show you another picture if I can. Check this out. They are small plants that are usually quite tiny. They consist of a stem and some leaves that are usually one cell thick or a couple cells thick. And unlike vascular plants, which are the seed plants, and the lycophytes, which are ferns and their relatives, they have their haploid phase is the dominant phase. So that's like the leafy part that you see. And they produce spores that are contained in capsules on a little stalk called seta usually. And so that's how they kind of get around. The spores blow all over everything. Here you can see this old truck that's kind of being colonized. They come in all kinds of shapes and colors. My favorite, sphagnum. These are the peat mosses. This is Sphagnum wolffianum. And let's just take a look at a liverwort. Here's a very colorful liverwort called Herbertus. So they come in yellows and blues and greens and all kinds of colors. What I'm hoping to do in my new position is to really delve into some specimen-based research, which is identifying new species based on collections that we have in the herbarium. I just published a paper about a week ago describing a new moss from Guyana, from the collection in the herbarium at the Smithsonian that had, there's so many specimens that are like just kind of in the backlog, what we call the backlog. So that's things that have never really been looked at very carefully. There's all kinds of treasures to be found there, and I'm hoping that we'll also be finding some new things to describe here. I love doing field work. Like I really love doing field work, getting out into the field and collecting new specimens. We might say like, we've already got over 300,000 specimens here, but a lot of them are, you know, they're getting older, and right now a lot of work is being done with like DNA analysis, molecular analyses. So we need to have fresh materials for that. Some parts of the province are very well collected, but others really are kind of unexplored. And so it'll be good in the next while to get up into those areas and get new specimens. Other things that I'd like to work on are just like outreach and education, like training the next generation of bryologists so that they can carry on the work that's being done by us older folks, I guess now. And just getting out, like once things are opened up a little bit, I'd like to be getting out and doing some moss walks and liverwort walks with people that they're just interested in, bringing them in to show them the collection, and just kind of raising the appreciation for bryophytes in general. And this isn't just, I guess I'm talking about bryophytes, but I'm sure that Mary Burby and Curtis and Trevor would also love people to be getting out and, you know, enriching our collections by doing more collecting and getting in and seeing what's going on around here with those things. One of my big areas of focus personally is in conservation. So in the last few years, we've had several species of mosses declared endangered by COSEWIC, which is a national conservation body of, you know, kind of composed of experts who take a look at what's out there and make recommendations to the government as to which species are at risk of extinction. And I'm also active with the IUCN on their moss committee, moss and liverwort committee. Anyway, that's a little bit about me. I'm sure that there are questions, so I think if anyone has some, I'll do my best to answer them.
Yeah, so Karen, I've got a great question from Ignacio, who is wondering on the topic of sphagnum, what makes it your favorite?
Oh, what makes my favorite? Well, for my doctorate, I studied peatland restoration. And so that sent me all over Vancouver Island and beyond, looking at peatlands, measuring different water quality parameters, water level fluctuations. It was in relationship with a potential restoration of a peatland around Victoria called Rithets Bog. And to do some kind of restoration, I didn't, at first I was really naive. I went out and was like, okay, yeah, we just need to do this and that. And then I realized, like, oh yeah, I know nothing about peat mosses. And, you know, each species, community has particular environmental conditions that they're associated with. And so I went around measuring all these kinds of things. Ultimately, I think the restoration of that peatland was kind of impossible because it had gone so long since actually being drained for agriculture. And it's really hard to just bring a system back in the current climate and whatnot. But as soon as I started delving into the different species, I came over to UBC and started looking through microscopes. And as soon as I got to see the detailed images of what the cell structures were like, it was just like, wow, these things are so beautiful. And so I really was attracted by the visual beauty of them. And now it's kind of partly like that kind of plant hunter type of thing where I'm like, oh, I want to see, you know, what's there. And it's kind of curiosity. So they're, besides everything, they're really, peat mosses, besides the beauty of them, they're really interesting plants because they hold so much water. They're really important when it comes to climate change with the things that are happening up north. I mean, there's just like a lot of different things with sphagnum that's quite cool.
Yeah, thank you, Karen. A lot of good reasons to like sphagnum. Sheila was wondering, have you been involved with Burns Bog as well?
Yes, I have been. I worked on the Burns Bog. I can't remember what it's called now. The review that was done with Richard Hebda, and wrote some pieces for that report, the Burns Bog Ecosystem Review. So I wrote the part on sphagnum and collaborated on some of the other sections of the report, which resulted in Burns Bog being protected as a park. And following that, I sat on the Burns Bog Scientific Advisory Panel for quite a few years. But I found it after a while, and living in Washington, D.C., it was a bit tricky to participate as fully as I would like to, but might get back to that now that I'm here.
Thank you. Ignacio and Sheila say thank you. Interesting comment from Arlene. And I didn't know this either, that there are endangered mosses.
Yes, there are quite a few endangered mosses in Canada. In 2016, I worked on an assessment for a really, really tiny moss. This moss would look kind of like the 5 o'clock shadow or 3 o'clock shadow or whatever time it is that men get with whiskers. It's so teeny, and you can kind of like look on the side of a rock and just kind of see it very barely sticking out. And so there was one species on Vancouver Island around Kennedy Lake out by Tofino and Ucluelet, and then also up in one of the inlets on the north end of the island. And that one is called Celigeria cutifolia. So it's a cute leaf limestone moss. So that one was thought to be, it was very rare. There were only 2 known occurrences from it. And a lot of these occurrences are known from our collections made by Wilf Schofield, who is like the major collector of the bryophytes that our collection is based on. And so that one was declared endangered first. And then following that, we assessed 4 species on Haida Gwaii. And all 4 of those have been declared endangered, one just recently. So if you're interested in seeing like any reports on these kind of things, you can find them on the COSEWIC website. So that's COSEWIC, Committee on the State of Endangered Wildlife in Canada. And you can search for the mosses there. So there are 4 there that were declared endangered. And then just now, like right now, we're just starting an assessment of the first liverwort, because liverworts have never been assessed in Canada. And it's not just these coastal ones that are endangered in the province, but there are also some elsewhere in the province in the interior as well, and even in the northern, or in the lower mainland. So, thanks for putting up that link, Nicole.
Yeah, so thanks, Karen. Yeah, Nicole was actually wondering, what are some key species that you could encourage new bryologists to learn for ones to ID in the field, or maybe some common ones we might see nearby?
Oh, there are all kinds. I think what would be really helpful, just to start with the basics, is to get a book like the typical what is the Andy McKinnon and Jim Pojar book, The Plants of Coastal British Columbia? And you can just pick that up, I think, pretty much at any bookstore, maybe even on the ferry or some gas stations or something, and take a look. There's some if you wanted to look at liverworts, you've got some really big ones that are quite common that could be found around, that look like a snake's tongue or a seal's tongue. They're kind of flat. These are called the thalos. They kind of like look a flat green ribbon. There are leafy liverworts. Oh yes, that the museum has this book's at sale. And then there's some pretty common mosses, like the pipe cleaner mosses, and I'm trying to think of all the common names of them. And step moss. This is Hylocomium splendens. It looks like like it has like 1 year of growth, and then another year of growth, and another year of growth on top, and you can count how many steps are in it to see how old they are. We've got things like big red stem, it's called, the common name, which is Pleurozium schreiberi. In fact, those 2 mosses, we're going to be doing citizen science monitoring, air pollution monitoring programs starting next year. It was going to start this year, where we've divided, with colleagues at Trent University, we've divided the entire country up into grid. And then we're asking people to go out and collect samples of these 2 really common, very easily recognizable mosses, and send them into the lab at Trent. And we're going to use them to measure air pollution concentrations throughout the country. So it's a way of getting involved.
Thank you, Karen. Speaking of, Sheila was wondering, when you referred to the peat bogs further north, were you referring to them in terms of potential danger regarding faster warming in the Arctic, regarding climate change, or?
Yeah, so peatlands store a lot of carbon, and it's all within the not just like on the surface, but the peat deposits can be quite deep. And then as climate changes and it warms, these things start to get dried out and decompose. So that carbon is potentially being released. It's also, when things are drier, they're catching on fire. And if they're not like super moist like usual, that's also being released. Another really cool thing about peatlands I forgot to mention is that they're quite acidic, and there's not a lot of decomposition below the surface. Like, there is some decomposition, but you can see, like, if you take a look down through time. You can see which species are found throughout time in succession down below. So you can see, like, it started out as this, like maybe even just some algae or something at the bottom, or some pond lilies or whatever. And then you can take a look. If you take the peat and you have a little sieve with very fine grid mesh on it and wash water through that, it'll leave some leaves in the sieve, and then you can take them and take a look under your microscope and see which species they are. You can actually identify them to species in many cases. They're preserved in there. So there's that's kind of neat too. And you can radiocarbon date the material and see how old it is and how old the peatland is. So lots of cool things about peatlands.
Cool. Thank you. Nicola is wondering, do we know all the species of the 3 groups occurring on the UBC campus? And do we have specimens for them all?
That's a really good question, and it's one that I really can't answer. But I know that Mary mentioned when I talked to her a few weeks ago that she does have students going and looking for mushroom specimens out around the campus. I imagine that for the bryophytes, we probably do have representative samples, but I don't know for sure for lichens, that's for sure. I'm just sort of starting to get into those collections, so don't have the answer for that yet. But one thing that we've been talking about that would be really cool would be to have really like photograph and have really nice specimens available that people can take a look at for each of the species in the province that we have in the museum.
Thank you. And when it comes to identifying them, Ignacio's wondering, what can you use to sort of differentiate between 2 different species of mosses?
Oh, I think Ignacio needs to come in and as soon as we can and check things out, and we'll teach him a little bit about it. But that would be fun. But generally, what we do is we have dissecting scopes and compound scopes. And so in a dissecting scope, we can take pieces of a moss or a liverwort or whatever, and take them and put them onto a little slide with a cover slip. So it might be just a leaf, or we might need to make cross sections from a leaf, or it might be a cross section from the stem. And we take those onto our compound scope, and we take a look at them at some higher power, and then we can see the cell structures. We can see, maybe the edge of a moss is a little bit, jig-jaggy because cells are sticking out the side. We might find that in this part of the edge, we've got like 2 cells thick. It might be a color thing. In liverworts, there are things called oil bodies, which are very pretty little structures that some of them are large, some of them are small. There could be a certain number of them. So we have all these ways of looking at them. And we also have what are called keys. If you don't know about keys, they sort of lead you down the path to what you're looking for. So it might say, does it have this? And if it's yes or no, and you kind of go through your key and key it out. We also, of course, nowadays, I'm not doing this because it's not practical for myself, but we also have DNA analyses where people are extracting the DNA from a specimen and analyzing it, and you can see that way if it matches up to something that's already known.
Thank you, Karen. Ignacio's wondering maybe not now, but where and when could they show up and help out with such IDing?
I think what we're hoping about to do, and what Olivia and I were just talking about yesterday, was it would really be nice to get some workshops going for the public and people who are interested to sometimes offer walks, and then also have people come in and learn the basics of how to do some identifications.
Thank you. Yeah, so Ignacio and anyone else interested, keep your eyes and ears out. You'll know. On the topic of when you're out hiking, Derek was wondering, speaking of liverworts and mosses, how can you tell a leafy liverwort from a moss?
Oh, that's a good question. So there are quite a few different things you can do to separate the 2 of them. But there are things like, for example, a moss will have the leaves usually all the way around the stem, whereas a liverwort they are coming out in from the sides, kind of flat. There are differences in the way the sporophytes grow. So in a moss, you'd have the sporophyte coming out from the tip or from the side. And the first thing that happens is the seta elongates, that's the stalk that it's on, and then the capsule is on the top. Whereas a liverwort, first the capsule forms and then it elongates. A moss has a stalk on its sporophyte that's very wiry and strong. The liverwort, it's very, very kind of ephemeral. It's like really delicate and kind of just there while the stalk is coming up. And then it just sort of disappears. So there's quite a few different things that you can do. We've got whole lists where we can divide them up. And if you have a book called some common mosses of British Columbia that was written by Wilf Schofield, it's got a list in there, or else you can probably find online.
Thanks, Karen. Speaking of liverworts, Linda was wondering where you could find a hornwort in the wild. What habitats to look for? Would you find them in the city of Vancouver?
I think most hornworts are here kind of found in drier areas. And what to look for is that the sporophyte on a hornwort looks like a horn. It's kind of like this long green or black kind of piece that could be like between maybe a centimeter and 3 or 4 centimeters that kind of elongate and then splits, and then the spores come out through that split. There aren't so many hornworts around. Like they're not as common as liverworts and mosses. So it takes a little extra looking, but they're on the Gulf Islands. They're in some dry spots, even in the interior of British Columbia, in the southern part of the province. They don't go too far north.
All right, thank you.
And they have beautiful spores. If you ever get a chance to take a look at hornwort spores under a microscope, they're just dazzling.
Yeah, Linda says, cool, thanks so much. All right, I have a question. Oh, Derek pointed out that Olivia found hornworts in someone's backyard here.
Olivia has a really good eye. She's made some really excellent finds.
So on the topic of hornworts, liverworts, mosses, we heard what got you interested specifically in sphagnum, but what got you interested in, well, bryophytes in general?
Well, I think sphagnum was definitely my gateway. I remember talking to Wilf Schofield when I was coming to UBC to do my work on sphagnum, and he would say like, "You're a bryologist, Karen," and I said like, "No, I'm a sphagnologist." And he said, "No, you're a bryologist." And so I think once I got beyond that, I started to look at other bryophytes doing surveys for rare species. And I think you just can't stop with one group. You just kind of keep on going. So then after I was really falling in love with sphagnum, there were all of the dung mosses, which are really cool. So these are mosses that have sporophytes that some of them are like an umbrella, and some are like a red umbrella, and some are like a yellow umbrella, and they grow on poop. And so, maybe spread by something like, or that's been put down by something like a moose or a deer or something like that. And flies come along and they're attracted to the poop. And these umbrellas have the spores in them. And as the flies are down, doing their thing, the spores fall onto them and then they fly onto the next place and they get spread like that from place to place on poop. But they're really beautiful. And so there's that. And then, like there's the liverworts, like the kind of reddish one that I showed the picture of is really cool. This one, let's see if we can do this again. I mean, how can you not find that beautiful? It's just really awesome. So I think there are places that you kind of go to and you're kind of like, oh, I wonder what's there, and I don't know. It's just pretty cool.
Yeah, thank you, Karen. So you have cool mosses. Nicole's wondering, are there any particular species of bryophytes that are on your your life list, the ones that you would love to see and maybe haven't yet seen in the wild?
Sure. One is Dawsonia. It's like there's a kind of common mosses around here that are in the like polytrichum. They're these kind of like, I think everyone's seen them. They're quite, tall, about, several centimeters tall, and they're very waxy and green looking. Well, there's some really, really tall ones that are like this big. They're like kind of like little mini trees practically. So I would love to see those. There's some in New Zealand that I would love to see, some other ones. Working at the Smithsonian, I've just been exposed to like so many tropical species that I would love to go and see. There are a couple that a friend there made me aware of that are in Colombia that I would like to see. So that here and there, but I just would love to go and see more things in British Columbia.
Thank you, Karen. Yeah, I hope you see them. Mary is wondering if she wants moss in her garden, what is a good way to establish some good ones?
I guess it kind of depends on what you're looking for. I know a lot of people are actually trying to get rid of the mosses in their lawns, which are quite nice. But I think generally it would probably be if you have kind of like a moist and shady spot in your garden, that's probably the easiest way to get them to establish. I know there's a whole book on moss gardening that was written by Annie Martin, which if you'd like, I could put a link to that kind of thing. There, of course, mosses grow everywhere, like even in deserts, but it might be harder to establish them. There was some kind of thing going on for quite a while. I heard like an urban legend or a rural legend or something, which was that if you took some moss and you put it in your blender with some yogurt, then you could like spread it around and that would get it going. But I don't think that really actually works.
Yeah, well, I guess we'll maybe try and experiment with that. Nancy was wondering about those taller bryophytes you mentioned. Do they have any special structures that allow them to be so much taller than our shorter bryophytes?
Well, we've always kind of said that mosses are non-vascular, so they don't have any tissue to conduct water up from the ground up to the top. But apparently there is some kind of conducting tissue in them. There also the leaves, when they're moist, they spread out, but when they're dry, they stay quite close to the stalk, and apparently water can somehow travel up. But I think the recent research, like just in the last year, has shown that there is some conducting tissue in that family, mosses in that family.
I guess what they say is in biology, every rule has that exception, right? So we have a question asking, what area of BC has the highest diversity of moss?
I think it would be the coast, and especially up on the northwest coast. There are so many species on Haida Gwaii. It's thought to be a glacial refugium, and it has so many species of moss, things that aren't found anywhere else in North America, and some nowhere else in the world, in fact. For example, there's, remember I was talking about the little like whiskery kind of moss, Celigeria. There's an endemic species, Celigeria cariana, on Haida Gwaii, and that's found at several sites, and it was named after the captain. It was named by Wilf Schofield and Dale Vitt, who was a bryologist at the University of Alberta, after the captain of the boat that was taking Wilf around to do a lot of exploring over the years on Haida Gwaii.
Cool, thank you. Annie has a question. It's actually, there's that saying that goes, moss always grows on the north side of trees. Is that true? And if so or not, why?
That's a good question, and I don't know the answer. I'm sorry. Something I have to look up.
Yeah, thank you. Well, I guess we can look into that. Arnold's wondering, any tips on looking for and gathering specimens? His uncle has a garden in the backyard and he's very interested in take a look if there's any. So any tips on looking for and gathering?
Sure. I think if you're going to collect specimens, the best thing to do is to try to make sure that you have all of one thing in each of your collections. You don't want to have like a whole mishmash of things, especially if you want to take it in and ask someone to help you identify it. It's a lot easier if you have like a nice clump. I'm sure there's a specimen around here somewhere. Let's see. Here's one right here. I was just looking in the collection at Sphagnum and found that something was misidentified just by chance. And so this kind of shows you, this is like a normal 8.5 by 11 piece of paper. And this shows you kind of the size of what we're looking for. A nice clump. You don't want to just pick like 1 or 2 little stalks, because if you do, then it's not really enough, especially if it has to be dissected. So it's good to, have at least like 8 or 10 stalks of moss. And so just take them and pick them out. And what you need to do is dry them slowly. So a perfect thing to collect them into is something like a paper lunch bag. And then you could just like put your specimen into the lunch bag and put it to the side of a room or something like that, and it gets nice and dry. You can kind of like flip it around every once in a while. If you can find some with sporophytes on, that will be very helpful for the identification, because they're also useful in some diagnostic characters. So put them aside like that, and you can then take a look in a book, which would probably give you like a basic ID for them, but isn't always accurate, because quite often you need to look through a microscope. But if you collect some specimens, then you can bring them in and or send them in, and we can help you identify them. Just don't send too many.
Thank you. Actually, yeah. So I was wondering, when it comes to preserving and keeping them in our collection for mosses, do most of them look like the example you just showed us?
The packets, yes, for sure. This is like our standard kind of packet. It's archival paper. It's 100% cotton rag non-acidic paper. It has a label which has the place where it's from. It's got an accession number, the identification, and then the longitude and latitude. We want to know exactly where it's from. We want to know who collected it, when you collected it, like which date, if you were with some other people, what kind of habitat it was in, that kind of thing. And so they're all dry. They're dried specimens, and yeah, just they're quite variable depending on what is in the packet, actually.
Thank you. Sheila was wondering, she's got her potted plants on a north-facing balcony, and the surface of the soils are packed with liverworts, she thinks. And she loves the biodiversity, but she is wondering, are the liverworts potentially depriving her plants of nutrients, or is it a nice, happy, symbiotic relationship?
I think it should be pretty happy. I don't know if it's symbiotic, but it unless they're like crawling right over your plants, I think it should be fine.
All right, thank you. Ginger was wondering if you had anything to say about the qualities of moss, for example, et cetera, their advantages, how they could be used, and maybe future potential for utilization by, I guess, humans.
I'm not sure I'm catching what you're asking me. Could you clarify a bit? Like, I mean, people use them for gardening. Peat mosses get harvested for gardening. It's kind of, I don't know if that's really a great thing. Ginger, could you give a little more detail?
Yes, I mean, I think what Ginger's asking, I mean, and I'm curious is, what are some ways that moss is very important for just the health of any ecosystem? You mentioned earlier being like carbon sinks, things like that.
Oh, sure. Yeah, I mean, like they're just part of the ecosystem. Quite often, like they'll be on decaying logs, helping break things down. They're helping retain moisture on the forest floor. They're being used by a lot of different species. I don't mean like necessarily for humans, but like if you're a bird or something like that making a nest, they're important for that. They're providing habitat for insects. I think they're part of the big picture. For us personally, I think that that's just important for us to conserve the biodiversity of all kind of organisms.
Thank you.
I see that Nicole is mentioning like that in the chat that peat is used for fuel. I think like as a sphagnum lover, I don't really love those kind of uses of them necessarily. So I think they have some important parts to play, but I don't know that necessarily like in the past, like there are definitely really cool ethnobotanical uses for like say for peat mosses that were used by First Nations people for diapers, for sanitary products or what do you want to call them. One time when I first came to the UBC Herbarium, one of the researchers brought back a whole bunch of sphagnum that she had been using as a pillow when she was doing field work in South America. So I guess they probably could have been used for things like that as well. But I think, I mean, just for us nowadays, I'm not sure.
Oh, no, thank you. Mary was wondering what we need to do if we wanted to contribute new specimens to UBC's collections. She'd love to know that some of her specimens will be there for future generations.
Yeah, absolutely. We're welcoming specimens, especially ones that are nicely collected and preserved. So, you know, if you're going to want to do some contribution of specimens, you can get in touch with me. My contact information is on the website, and I can give you a spreadsheet of the type of information that we appreciate receiving with a specimen, because it's not really helpful if you send in, you know, mushroom from backyard, don't remember when, or that kind of thing. Like if you give us the basic data, like the type of thing I was mentioning on the labels, we can give you some instructions on how to properly dry and prepare your specimens, what information we need, and then definitely we'd welcome receiving them, especially if they're things that aren't like super common. So if it's something that you see around all over the place, it's a little less interesting than things that are a bit less common, but definitely feel free to get in touch. I would be happy to receive specimens, especially like you've heard that for the Bryophytes, we have over 300,000 specimens, but we have a lot fewer lichens and even fewer fungi. I think also, like, if you're interested, we can probably give you some contact information for different groups. There are different groups focusing on these different groups of organisms, like, I mean, groups of people that you might be able to connect up with in the community, besides going on any offerings in the future that we might have.
Thanks, Karen. Yeah, we'll keep that in mind if we ever do some collecting. Laura's wondering, when do bryophytes release their spores? Do they have a similar life cycle to vascular plants? For example, blooming in spring, summer, and releasing seeds summer, autumn?
Yeah, things are happening all the time. So very early in spring, some different species are releasing their spores, and all through the summer. And then some peat mosses, interestingly, are their sporophytes are bursting forth, like they have for peat mosses anyways, they have like a little cap on the top, and they pop right off, and spores are spreading, and that can happen all the way through September. So all spring and all summer, and then into the early fall as well.
Oh, all right, thank you. Ginger was wondering, are there any mosses you can eat?
Mosses aren't really, they don't have any nutritional value. So I don't think it would hurt you to eat them, but they're not going to be really doing anything for you. Although like again with sphagnum, I was told that the sporophytes, the capsules are very peppery flavored. And so I tried them, but it's like, yeah, not much there.
Noted. And Nicole did mention, as far as human use for it, not quite eating it, but it is used in the production of scotch.
Yes. Oh, yes. That's very important. Right. To flavor the smoke that makes the flavor of the scotch. Yeah, definitely. Big time.
All right.
A good human use. I'm glad you thought of that, Nicole.
I had a question. If there was one thing you wish people understood about moss, fungi, and lichen, what would it be?
I just wish that people like knew more about them, like and how diverse they are, and and they're just like so much of like the richness of our province in British Columbia and all over the place. Like you often quite like could overlook things, you know, like you're just looking at the beautiful flowers or something like that, or the big trees. But once you really start to get into the details and start to appreciate these things, you can't kind of unsee them again. They're just so nice. Like I had a volunteer and an intern when I was working at the Smithsonian, and both of them like they came to help fold packets and things like that. It's a little bit, you know, boring after a while, even if you like doing something like origami. It's just like, hmm. So giving them opportunity to take a look through the microscopes and get into the specimens, and then they were both like, wow, we just had no clue. And now whenever we go out with friends, we're like, hey, there's a moss and there's whatever. Like they're just really, their eyes are open, because at first you're kind of like, oh, it's just all that green stuff. And then once you really get to learn a little bit about them, you realize like, wow, they're like in trees and they're in cracks in the pavement, and you're out in a really dry spot and it rains. And it's like, oh wow, look at that. I didn't think they would even grow here. I thought they would only be, you know, in places that are super moist and lush, you know. So I think it's those are a couple things.
Yeah, keep that in mind. Yeah, I'll admit maybe I don't pay attention enough to moss. I'll definitely keep an eye out for it.
This morning on Facebook, I saw a friend of mine posted this video, which I also reposted, and it was this bird, and the bird, it was like a male bird of paradise, and it was, you know, dancing around and cleaning up its little space where it was going to like show off, and then it started to dance once this female bird came along, and you know, it had like these beautiful like kind of pom poms sticking out of its tail, and it flashed yellow spots and all this kind of thing. And I was thinking, man, it's really hard to compete with some of those things, you know, the charismatic megafauna and the beautiful birds and things. But yeah, there's something to be said for the non-charismatic microflora as well. You know, like we've got some pretty cool things if you just actually take a chance to look at them closely.
Yeah, thank you. Uh, uh, so you've just recently come back to Vancouver, and you told us a little bit earlier, but uh, what is your, tell us a bit more maybe about your connection to the UBC Herbarium, like how it's been a part of your kind of career and in the past and moving forward.
Sure. So quite a long time ago, I did my undergrad at UBC in landscape architecture, and for my final project, I did a restoration project of a pond in Victoria called King's Pond, actually in Saanich, with Nancy Turner. She's an ethnobotanist. And I worked with her students at UVic to do the pond restoration. And then after working as a landscape architect, when I graduated, I found that just wasn't really, I wasn't doing what I'd hoped to do, which was something more to do with restoration or something like that, ecosystem restoration. And so she asked me to come over and be her graduate student. She asked if I'd like to do that. And I was really excited and thought, yeah, this is awesome. This is perfect. So I came back to UBC and did a little bit of upgrading of my biology courses and started over with her. And then while I was working on my grad work with Nancy, I started coming to UBC because it's got such an excellent collection. And Will Schofield was here, and he was, you know, like our #1 bryologist. And so I did a directed study with him, and I really just loved hanging out in the herbarium back when it was in the biological sciences building up on the 4th floor, I think it was, and spent a lot of time there working through the collections, and made a lot of nice friends with Olivia and Wilf and Terry McIntosh, and it was just really welcoming. Patrick Wilston. And so I got involved then. And then after I graduated, I spent some time doing these kind of rare plant surveys and working with the British Columbia Conservation Data Center, helping them do some ranking of mosses and liverworts, and became a member of the Mosses and Lichens group in Kosiwik. And so all of this work involves specimen work, looking at specimens. So that was kind of like my major connection. And then at some point I went to do a postdoc in the Smithsonian. and my project that I was working on there, the funding ran out after the first year. But in the meantime, I'd been going to the herbarium there to still identifying specimens, processing specimens. My mom is just like, when are you going to get those specimens out of my house? And so they asked me, would you like to come and work on our backlog of bryophytes and help distribute it? It had been really like building up over the years on the top of the cabinets. And then they decided we need to rearrange our collection. And so I work on the rearrangement. And all this time I've been having a connection with UBC because one thing that herbaria do is that they exchange specimens. And so the Smithsonian was like, you know, we've got like an extremely large debt owed to UBC. And so why don't you prioritize sending some of our excess specimens to UBC? And all over the years, I've been coming back to UBC. I'm an honorary research associate in the Department of Botany. And so as I've been doing different reports, they require specimen work. And here we have the specimens. And then just recently I got hired here to continue on the work that Olivia's been doing. So that's kind of been my connection with the herbarium. It's been really great. Everyone here is so welcoming, and yeah. And I've been doing just little projects here and there, all of them making use of our collection at UBC. So super excited to be back here.
Thank you, Karen. Yeah, we're excited to have you back as well. So many great connections. Thank you so much for sharing that. Did you have anything else you wanted to say to anyone, or if did anyone else have any final questions, you can put them in the chat.
I just want to thank people for coming. I'm really excited. I saw some friends in the chat earlier. I can't see everyone right now, but I saw quite a few people that I know, and I really appreciate you coming. I kind of came here in a really weird time because it's been this COVID time, and so kind of left my people in DC and came across the continent with our rental truck and all that kind of thing, and haven't really been able to get in here as much and welcome people as much as I'd like to in person. But once things get going again, we'll definitely see who can come around. And
yeah, big thank you from everyone. One last question for Nancy, a bit of a biodiverse going across phyla. Do any of our moss specimens, when maybe looking at them under the microscope, have any tardigrades dormant amongst them?
There are tardigrades in specimens. I haven't noticed any when I've been looking at things in the herbarium, but definitely I have things that when I've been in the field seen tardigrades. Yeah. So if you want us to keep an eye out for that, definitely we'll do that.
All right, thank you so much. Everyone is sending their thanks. It's very enlightening. Thank you so much for your informative talk. Thank you so much, Karen. I do want to just thank everyone for your support, supporting us here at the Beaty Biodiversity Museum. Thank you to those of you who donated online when you were registering. And of course, if you're joining us for future Beaty at Homes, we appreciate that, and if supporting us continually. And yes, next week, our session is Eco Innovations Inspired by Biodiversity. So that is next week at 1 o'clock, as per usual. You can find the link at beatymuseum.ubc.ca/events/event. So connect with us online at our social media. So just at Beaty Museum, and here are the links if you wanted to find more about our online resources. But once again, we wanted to thank everyone for joining us. And of course, to Dr. Karen Golinski, thank you so much. And thank you to everyone for joining us for one more Beaty at Home. And yes, thank you everyone for being a part of Beaty at Home, and have a great day. Thank you so much.